So I have to share my presentation, and while the presentation is open, I'd like very much to thank the organizing committee to invite me to share our data about autophagy in the mechanism involving the development of diabetic retinopathy. So may I share the screen?
Yes, please.
Everybody seen my presentation?
Yes, if you could just put it in the full screen, that would be better visualized.
Excuse me, which mode of presentation you prefer?
I mean the full screen one.
The full screen one. Let's see. I cannot see this. Why? No. So is there anyone who can help me, because I cannot see? Share is paused. Stop sharing. I don't know why I am not able to.
I think there is in the bottom right side there is like shared or full screen option.
The right side. And share. Why I cannot share the? Are you seeing my presentation?
No problem. Yeah, it's seen. Yeah, clearly, no problem.
You are seeing.
Yeah.
Is that correct?
Yeah.
But it's not as the one that you like to do.
Yeah, it's okay, but just to be more visualized. Otherwise, it's clearly seen now.
So we are going to talk about autophagy or deficiency in autophagy in the development of diabetic retinopathy. Again, thank you for the invitation for this nice and very big conference. As everybody knows, diabetes is a global emergency around the world. Today, we account with around 400 million people living with diabetes. And most of the projection shows increasing this number, reaching 600 million people in 2040, which is a really global emergency. Diabetic retinopathy is one of the chronic complications in diabetes. It reaches about 70% of the people around 12 years of duration of diabetes in different levels. Still, diabetic retinopathy is the major cause of irreversible blindness in the productive age of the people, so it's a very devastating complication. And here on your right side, I'm showing you a retinography of a patient with diabetes, and he's presenting a very exuberant macular edema with a lot of hemorrhage and exudates in the retina, which dropped down pretty much the vision. About the autophagy. Autophagy is a mechanism very well preserved and evolutionary preserved, and many regulated by different stresses and stimuli, such as infections, cytotoxicity, starvation, hypoxia, endoplasmatic reticular stress, turnover of long proteins, curious of aggregate inside the proteins, suppression of apoptosis, nutrient recycling, leading to either cell death or cell survival. So the main goal of this very well-preserved mechanism is to maintain the cellular homeostasis. The whole sequence of the events are orchestrated by about 35 or 40 different genes. Initially, damaged organelles, proteins, these malfolded proteins, pathogens which are inside the cells. They stimulate the endoplasmatic reticulum stress response, which will produce fragments of the cell membrane. This the P62, which is a cytosolic protein, will bind to this ubiquitinated protein, recruited by LC3B, which is another cytosolic protein. And this will be all organized. This is in a very simplified way to explain this. This will be all organized to form an autophagosome structure. The autophagosome will fuse with the lysosome, which has the function of a proteolytic enzyme with its proteolytic enzymes, and will liberate the LC3B to return to the cytosol, degrade the content, and clearance with amino acids, fatty acids, and so on, reestablish the energy and the recovery the energy, the hemostasis of the cell. Autophagy is not only present in diabetes, which is not very well known, but is present in many other diseases, neurodegenerative diseases like Parkinson, Alzheimer, also type 1 diabetes and juvenile arteritis, inflammatory disease like bowel disease, and also in cancer. I'd like to highlight the retinal Miller cells. Miller cells is the predominant glial cell in the retina and interact very well with most of different cells, meaning neuron and vascular cells. Diabetic retinopathy today is accepted to be a neurodegenerative disease in its early phases, with vascular changes in its late phases. And this Miller cells is a very complex cell with many functions, such as releasing trophic factors, recycling neurotransmitters, controlling ionic balance in extracellular space. And under stresses like a high glucose condition, Miller cells react as change the morphology, the physiological and biochemical as a reactive gliosis. And also one of the changes is the overexpression of VEGF, which is a very well-known therapeutic target for pharmacological treatment for diabetic retinopathy, either to treat macular edema, as in that picture that I showed in the beginning, or in its proliferative phases, advanced proliferative phases. So the aim of this study was to investigate whether autophagy is playing a role in the cellular and molecular mechanisms involved in the diabetic retinopathy. For this, we study glial cells or Miller cells in normal and high glucose conditions, mimicking diabetic Miller, with or not treatment, like pharmacological activators, inhibitors, and silencer RNA. Starting, we observed that cells under high glucose conditions exhibit increasing in Becklin 1 and LC3-2-1 ratio, which is a marker of initiation of autophagy, and increasing of this P62 sequestrosome, which is a marker of the cargo inside the autophagosome, meaning that there is activation of the autophagy in the beginning, with activation of the endoplasmatic reticulum stress response, seen here by increasing of phosphorylated F2 alpha and SHOP proteins. Also, increasing in caspase 8 activity, which is a biochemical marker of apoptosis, one mechanism of cell death, and increasing in cell death marked by tunnel assay. in high glucose conditions. In this scenario, we decided to inhibit the autophagy, since the autophagy initiated by high glucose was damaging for the cells. So we treated the cell with 3-MA, 3-methyladenine, which inhibits the fusion, the initiation of the autophagy. Here we see also in normal as in high glucose, the 3-MA decreases the initiation of the autophagy, but worse in another sense, the accumulation of the P62. And the P62 interacts directly with caspase 8, which is a biochemical inducer of tunnel, which is the marker of apoptosis. Next, we silenced the P62 protein in order to ameliorate the situation. So the silence of the P62 was efficient, we can see by 60%. And this, it decreased in the high glucose condition, decreased the expression of phosphorylate PF2 alpha, which is a marker of endoplasmatic reticular stress. Oh, sorry. Decreased the caspase 8 activity, decreasing apoptosis, marked by tunnel assay, which was a protective effect, the silencing of P62. Then there are many stimuli upstream to stimulate or inhibit autophagy. One of those is the rapamycin, which blocks mTOR, activating AMPK, thus activating autophagy. In this set of experiment, we treat the cells under high glucose with rapamycin, and we could see clearly the increase in Becklin, which is indicative of initiation of autophagy, and a decrease of P62 sequestrosome, meaning that all the content was properly digested by lysosome, decreasing the activity of caspase 6, preventing the apoptosis. To better understand how rapamycin could improve the autophagy flux in cells under high glucose condition, we put, is not a treatment, but is a tracer, acrid in orange, which markers organelles in very low pH. So in the normal condition, we see in green the cells, the autophagosomes, which are fused with the orange organelles, which are lysosome, and this leads to a proper digestion of the content. In the high glucose, by contrast, we see a lot of autophagosomes with very few lysosome fused and functionally. In the presence of rapamycin, high glucose cells under high glucose condition return, or rapamycin restore the activity of lysosome, similar seen as in normal condition. This is why, because the lysosome, rapamycin ameliorate, upregulate the activity of many, of at least one of the most important proteases, lysosomal, which is cathepsin L. There is an increase in cathepsin L activity in cells under high glucose treated with rapamycin. And this, as a proof of concept, we see that the production of VEGF which is one of very, very well targeted to control pharmacologically, the whole process is significantly decreased. And this is also true, not only in cells, but also in retinal tissue. When in the control animals, we see the expression of Becklin because there is a normal turnover of organelles and other debris inside the retina through autophagy, but this is pretty much increasing in presence of diabetes, experimentally induced diabetes. And in also these animals, the expression in red of P62, the content of autophagosomes, were also accumulated compared to animals, control animals, not diabetic ones. And also in animals, not animals, but in retinal tissue, there is a decrease in cathepsin L lysosome protease activity. So in summary, I'd like to say that diabetic or diabetic Mille condition inducing post-translational changes, which change the folding of the proteins, stimulate the endoplasmatic reticulum stress, which will initiate the production of the isolated membranes and formation of autophagosome, which will be fused to lysosome, but due to a lysosomal dysfunction, this cargo is not degraded. There is a direct interact with caspase 8, leading to higher apoptosis. In conclusion, the appropriated autophagic process is an important mechanism of cell homeostasis in diabetic conditions. The improvement of lysosomal activity may be of therapeutic value in disorders by which autophagy lysosome degradation plays an important role. I'd like to thank you, FAPESP, to support our research, our team, and thank you, everyone.